Viruses in general have common patterns or common ways that they will release from their infected host cells. We're going to find that for both prokaryotic and eukaryotic cells that are infected with lytic viruses, the lytic cycles are very similar. And of course, the endpoint is the lysis of the cell that was infected and was the place of the assembly, biosynthesis, maturation, and release of the new viral particles. When we found that temperate viruses for bacteria could undergo prophage formation in lysogeny, or during the lysogenic cycle, scientists wondered, can we find eukaryotic versions that are similar to what the temperate phage does in prokaryotic cells? And sure enough, they did. We do find that there are eukaryotic viruses that will infect animal cells and form provirus formations, which means we have viral DNA that has been inserted or fused with eukaryotic chromosomal DNA. And we'll have more specific examples of those when we go through the specific kinds of diseases caused by viruses. And then a third possibility that's unique to animal cells are animal viruses that cause infections, but often are described as slow infections, where we essentially have the virus gaining entry, slowly beginning the process of causing the host cell to dedicate its cell function to the production of viral proteins and nucleic acids, and then emerging from the infected cell, not by cell lysis, but instead little packets that we call buds. Now, this budding release is sort of visually similar to the formation of budding or buds by yeast. They're not exactly the same, but you'll see in the illustrations, these are just tiny little packets. For viruses, they will be the complete final assembly or structure of a new viral particle, if the virus is releasing by budding. Here's a close-up example of how a virus, especially an animal virus like HIV, attaches to the host cytoplasmic membrane. We call it receptor-ligand interaction. There are viral spikes sticking out of the surface of HIV that will specifically attach to structures sticking out of certain types of cells in the human body. It's very much like a hand in a glove. When the viral glycoprotein spike attaches to the host cytoplasmic membrane protein receptor, the receptor and the spike change shape a little bit, but what's most important is the change in shape of the host receptor when it locks or when it binds with the viral spike, and that mechanical structural change is essentially translated through the cytoplasmic membrane into the host cell, and almost like a garage door opener, the host cell starts to transport the virus inside. It's analogous to a lock and a key, or I think garage door opener is a little bit better, because there's so much flexibility and sort of bending and changing of shape when the viral spikes lock onto the membrane protein receptors. They're not rigid structures. They're very flexible. And it's the flexing or the mechanical change in shape that activates penetration of the virus into the uninfected host cell. Here's an example of a budding virus. HIV is one of those types of viruses, the human immunodeficiency virus. It's a eukaryotic virus, and it's going to essentially gain entry into a cell by attaching or adsorbing to the cytoplasmic membrane, again via specific receptors. the whole entire envelope of the virus fuses with the cytoplasmic membrane, dumping the capsid containing the nucleic acid inside of the cytoplasmic membrane. So if we notice at the top of the illustration, the viral envelope fusing, the capsid penetrating, and notice in this particular illustration, we're going to find that even though this is an RNA virus, we're going to be able to, if we are this type of virus, produce a DNA copy of the RNA, which in fact becomes inserted into the eukaryotic host cell chromosomes, forming a particular new construct, similar to the prophage, only this is, of course, a eukaryotic cell. Now the new viruses and the new genetic material directs the biosynthesis of viral proteins and nucleic acids, typically in a slow, progressive process, often described as a slow infection. So unlike a lytic infection with essentially fairly rapid production of new viral particles, with a slow infection, like one with HIV, in fact, we have sort of a slow gradual production of new viral particles. We essentially would have release of new viral genome, the production of new capsids, packaging everything inside, and as this newly assembled capsid goes through maturation and now is aiming or heading for the outer world for release, notice that we have essentially three places or three ways that this virus can attain or receive its envelope. One place that the newly formed virion or viral particle can gain its envelope is the host cell membrane. That's labeled number 1 near the bottom of this illustration. Another place that this envelope virus could possibly attain its envelope is the nuclear membrane. The third possibility is that there are membrane structures floating in the cytoplasm and it's acquired there. Now, typically for HIV, it's the cytoplasmic membrane is the source of the envelope for HIV. But regardless, these are still very similar types of structures, phospholipid, biological membrane derived viral envelopes. Here we have an example of a naked virus that causes infections in human cells, the papovirus. The papovirus causes papillomas or warts. Notice that with step number 1, the virion attaches to the host cell, it penetrates and then releases its viral genome, which directs the synthesis of viral DNA and viral proteins. We have assembly, maturation, and release. So very similar to the naked viral lytic cycle for a prokaryotic cell, here we have a eukaryotic animal virus undergoing a very similar kind of infection and growth cycle process. Here we have a more generalized example of the fusion of an envelope virus with a host cell, the release of viral DNA with the formation of a provirus, which means that the viral DNA becomes fused with the eukaryotic chromosomal DNA, and then every time the chromosome undergoes replication, the provirus is also replicated. But in the meantime, the viral DNA is also directing the production of viral proteins, viral nucleic acids, and again, the slow release by budding of new assembled mature viral particles as illustrated in step number 5. In this illustration, the acquisition of the envelope is from the cytoplasmic membrane. In this illustration, we see two possibilities for entry of an envelope virus into an animal cell. The most common is in the first column, where we have fusion of the viral envelope with the cytoplasmic membrane. It's receptor mediated, but we'd only dump out or penetrate the host cell with a capsid containing the viral genome. In the second column, it's also receptor mediated, but notice when we lock into the receptors, it causes essentially an extension of the cytoplasmic membrane, so the entire viral vesicle forms, where we bring in the entire construct, envelope, capsid, and nucleic acid. Once inside, this unique vesicle that contains the enveloped virus is dissolved or opened up to release the capsid. So penetration is a little bit more lengthy and involved process in this type of what's called endocytosis, where we're bringing in the entire structure of envelope, capsid, and nucleic acid. Here again is a close-up version of where viral spikes come from, or at least an illustration of one way. Notice at the top we have a fully assembled capsid with the nucleic acid inside. There are viral spikes that become inserted into the cytoplasmic membrane, and as the capsid emerges through that membrane structure, it is now thoroughly modified with viral proteins in the form of viral spikes. Once completely released, in this process that we call budding, we now have a fully formed and fully assembled enveloped virus.
The photo at the top middle of this image is a color-enhanced transmission electron micrograph. TEMs, or transmission electron micrographs, come in black and white and various shades of gray. But to enhance the image of the virus, artists will color in little bits and pieces so that students and scientists get a better idea of what they're looking at. And you can just see at roughly a 3 o'clock position on the cell that's releasing the virus, there's just a little pink bleb right here, indicating the emergence of a newly formed viral particle.
Now, how can we tell if a virus is infecting a cell or if someone has a viral infection? Well, there are two ways or two methods that can be used to test for the presence of viruses. One would be in vivo. In vivo simply means in an animal or in essentially a living organism. In vitro means under artificial conditions in the laboratory. Let's say where we're using growth medium in a test tube or a petri dish. So in vivo, using an animal as a test model. In vitro, using something in a plastic or glass dish or a test tube. Here's a common way of cultivating viruses in vivo. Now, decades ago, we did use animals. In fact, we can still today use lab animals if we need to. Take a specimen from a patient and inject it into an animal to determine if a virus is present. Decades ago, we really did use guinea pigs because they had turned out to be excellent models when we were trying to study diseases like viral infections. More likely today, we'd be using fertilized, embryonated chicken eggs. Notice in the illustration, there are various membranes and cavities in this embryonated egg in which we can inject a sample and then days later go back and retrieve a sample to determine if a virus is growing either in one of the egg cavities or one of the specialized egg membranes. Egg cultures are extensively used in the production of vaccines. If you've ever heard on a commercial that if you're allergic to eggs or birds, you cannot take a particular vaccine, this is why. Because there are literally hundreds, if not thousands, of eggs that are used every year in the manufacture of vaccines that are used to prevent diseases or infections. Just a very good model and a very good production product. Now, in vivo techniques are fairly technically challenging, aside from the fact that most of us, including myself, really don't like to have to use animals. It takes longer sometimes to get your results, and of course there's the problem of housing them. It's very expensive, and I just don't like sacrificing or killing the animals if we have some good alternatives. And sure enough, thank goodness we do. In vitro cultivation of specimens to detect the presence of viruses are now very common methods and very cost-effective methods. For a sort of a very reasonable process of in vitro cultivation, we use tissue cultures. The tissue cultures are, as the name implies, simply cells from tissues in an organ or from an organ in an animal's body. We could take the tissue from any particular body site, mince it up with scissors, and this is all done aseptically, so everything's sterile. There is no contamination with bacteria, fungi, or any other microorganism. We mince those tissues up and then add an enzyme like a protease to separate the individual cells in those little bits of tissues, put them in a test tube, a flask, or a petri dish, add some liquid growth medium, and allow the eukaryotic cells to grow. When we look through the microscope, as in this illustration on this slide, what we should see would be a nice confluent, which means we have one cell touching another, monolayer, single layer of eukaryotic cells. Now, there are various types of tissue culture cells that are used. We can have primary cell cultures. A primary cell culture means that we've taken the tissue from an animal, we've put it into culture, in other words, we've minced it
and chopped it up into little pieces. And then we use some enzymes to separate the cells so we can create a nice continuous single layer of eukaryotic cells. Once we have that nice monolayer of cells, we can add our patient sample and look for cytopathic effect, or the presence of virus as it changes normal cell function into the virus manufacturing factory that viruses can do to eukaryotic cells.
So here we have our tissue culture in a test tube and then into a petri dish. Typically the growth medium that we use for tissue cultures are liquid. And then we add our specimen that may or may not have a virus. If the virus is present, it's going to cause, of course, a pathological or change in the normal function of the cell. We call that a cytopathic effect. And CPE, cytopathic effect, can often be visualized or seen. The most frequent kind of CPE are the formations of plaques. We see plaques as essentially a moth-eaten cell culture. Plaques are produced by lytic viruses. In other words, this nice monolayer of confluent cells that we should have in our tissue culture, when we look at it the next day after we've exposed it to our potential source of virus, some of the cells are just gone because they've been lysed. So that the confluent layer of eukaryotic cells just isn't there anymore. We have spaces or empty spots on the petri dish where the eukaryotic cells have been lysed or destroyed by the virus. We can also see inclusion bodies, so that if we take the tissue culture cells, stain them, and look at them through the microscope, we can see structures in the cytoplasm of the cells that weren't there before. Again, the inclusion bodies are visible evidence of viruses growing. Now, it's not the actual virus, but they're clusters of materials in the cytoplasm of the infected cell that are large enough that when we stain them, we can see these evidential or apparently different structures in the cytoplasm. And then the third possible CPE that we might notice is transformation of the tissue culture cells from essentially normal cells to what are called transformed cells. And that's what's illustrated on the cartoon drawing at the bottom of this slide. The normal cells are on the left, and notice that they form a nice, even monolayer on the bottom of the petri dish. Transformed cells, and not all viruses will do this. In fact, viruses in general will not do all of these types of CPE. Typically, viruses will only do one of the various kinds of CPE that we can find in nature. But cell transformation is the one that often kind of troubles us the most, because a transformed cell is one that has lost control of its cell growth. Normal healthy cells have the ability to what's called lose its ability to undergo mitosis when it touches another eukaryotic cell. It's called contact inhibition. Contact inhibition means that as the eukaryotic cells undergo mitosis or cell division, and they grow, and when they touch another cell, it will trigger those cells to stop mitosis. With certain types of viruses that infect eukaryotic cells, those viruses cause a loss of contact inhibition. In other words, the eukaryotic cells just keep growing and growing or undergoing mitosis, and they just keep piling up in little blobs or little sort of collections of cells piled one on top of another. Well, what does that remind you of when we're talking about different pathologies or diseases that affect, let's say, people and animals? Even plants can have these things occurring. Typically, students usually realize that this is very similar to what we see in the formation of tumors. Tumors especially often have that loss of control of growth. They just keep growing and growing and growing, and oftentimes it's due to the fact that a virus has triggered the loss of control of cell growth. So all of these are examples of cytopathic effect, ways that we can see that viruses have been present and have caused some type of pathology in the infected host cell. Here we have an example of viral plaques in a bacterial culture. So remember, viruses can infect prokaryotic cells, they can infect eukaryotic cells, plants and animals. I really don't know of any cell on Earth that doesn't have its own unique virus that can cause its problems. Now, notice in the two photographs, the one on the right is, of course, a low magnification. The one on the left is a much higher magnification. And all of those little sort of dots that we see on both photos are plaques. These are actually empty spaces in a confluent lawn of bacteria. By confluent lawn, we simply mean these are layers of bacteria that cover the entire surface of an agar plate. And then when we introduce the viruses that are lytic, of course, because these are prokaryotic, we have a cell wall or peptidoglycan layer that prevents the viruses from budding out, so we have to undergo cell lysis if we are a virus that's infecting a prokaryotic cell. And ideally, every plaque or every little dot represents where one virus infected one bacterial cell released new viruses which infected the neighboring bacterial cells. So you get this moth-eaten effect, or all these little holes in the confluent lawn of bacteria. We call each of those little holes a plaque. And by counting the number of plaques, we get an idea of how many virus particles were in the original specimen.